Hello, my dear students and friends. Welcome back to Sanhita Chatterjee's biology classes. And today we will learn about protein because it's a very important component of our body, so that it is known as the building blocks and it makes us more strong, healthier person. So let's start. So, what are proteins? Proteins are large, complex molecules made up of amino acids and which are often referred to as the building blocks of life, and chains of amino acids linked by which bond peptide bonds, and they play countless critical roles in the body because we will see the functions of proteins. But before going to that topic, see what are peptide bonds. Peptide bonds are the bonds that get link the amino acid residues with one another. Okay, as if we know that glucose, and when two glucose attached with each other, it forms maltose, right? So glucose are attached with glycosidic bond, whereas amino acids attached with peptide bonds and forms proteins. Okay, so in the next slide, here's the listed functions of proteins. Structural function that build muscles, skin, hair and nails. Example, collagen and keratin. Enzymatic functions, so it speed up chemical reactions and digestive enzymes. Transport, it carries substance like hemoglobin, it carries oxygen, transports oxygen. Hormonal proteins, some hormones are proteins, example insulin. Defense, antibodies that fight infection are proteins. And storage and movement, store nutrients and help muscle contract. So we are having contractile proteins. For that we can contract, we can that finger movements and your every type of muscle movements, contraction and relaxation, it cause because of we are having contractile proteins. Okay, but most specifically in today we will study about its digestion and absorption of proteins, not about its structure and function. So proteins digestion start within stomach. Okay, whereas carbohydrate digestion only take place in mouth. If you want to know about the carbohydrate digestion, I have a separate video in my channel. You can go and check that one also. Okay, so where digestion starts? Protein digestion starts within stomach. Stomach is having so many type of cells. Okay, within stomach there is parietal cells, and this parietal cell secretes most importantly HCl or hydrochloric acid. Acid means it's having lower pH, right? So it denatures proteins, it unfolds them to expose peptide bonds. That is why stomach secrete one enzyme which is pepsin, but an enzyme secreted as inactive pepsinogen. So firstly stomach secrete pepsinogen, which is the inactive form of pepsin. Pepsinogen is secreted by stomach, which is the inactive form of pepsin. But this pepsin get activated when it get in touch with hydrochloric acid, HCl. Okay, so who activates pepsin? That is HCl. And this pepsinogen is secreted by Chip cells, that is another cell present within stomach. So Chip cell secrete pepsinogen and HCl is secreted by parietal cells. Okay, activated by acidic environment, breaks long protein chains into shorter polypeptides. So what is protein and what are polypeptides? Polypeptides are the amino acids, when less than 100 amino acid present, then it is known as polypeptide, whereas more than 100 amino acid, if get linked by peptide bond, then they known as proteins. Okay, so what happens here, basically these are the chain of amino acids and these bonds are the peptide bonds. These bonds are the peptide bond. So this pepsin, it is the activated form of pepsinogen. Okay, it cleaves these peptide bonds and make it shorter polypeptide chains. If it is 2 amino acid is present here, it is a dipeptide. If 3 amino acid present, this is dipeptide, this is tripeptide. Okay, and if 2 or more, then it's oligopeptide. Okay, now the most important part of our digestive system, that is pancreas. So after stomach, firstly it was stomach, and stomach secreted inactive pepsinogen, which get active pepsin, its active form, by the action of HCl. Okay, now this digestion move forward and it is within the pancreas. So within pancreas is the house of enzymes, because it's known as the mixed gland, as it secretes the enzymes as well as hormones. So pancreatic enzymes are known as zymogens involved, that there is the zymogens, name of the zymogens are trypsinogen, chymotrypsinogen, procarboxypeptidase, proelastase. Okay, so here pepsinogen, trypsinogen, that gen means the inactivated form. Okay, and these pro means also the inactivated form. Okay, so trypsinogen to trypsin is the active form. Chymotrypsinogen to chymotrypsin is the activated form, and procarboxypeptidase to carboxypeptidase A and B, these two get activated. Proelastase to elastase, it is the activated form. But these enzymes remain in inactivated state within the pancreas. Otherwise, what happen? Otherwise, it will digest pancreas also. Get it? So, site of enzyme action. Although the enzymes are secreted by the pancreas, protein digestion occurs within the small intestine. So protein digestion cannot occur within pancreas. Pancreas only secretes the important enzymes that can cleave a long chain of protein into its splitted small particles. Okay, so where is the site of enzyme action? That is the small intestine. Within small intestine, protein digestion can take place, but pancreas will secrete all the important enzymes that will act on the small intestine. Okay, so where these enzymes become active? See, we classify enzymes that are secreted by the pancreas in two groups, that are endopeptidases and exopeptidases. Trypsin, chymotrypsin, okay, trypsin, chymotrypsin, elastase are endopeptidases. They cleave peptide bonds within the protein chain. But carboxypeptidase is an exopeptidase. It removes amino acids from the end of the peptide chains. How it occurs? See, if it is a chain of amino acid, okay, so the amino acids are the circles. These are the amino acids, and they are joined or linked by the peptide bond. So these peptide bonds are within, because here is the amino acid, here is the amino acid within the peptide bond. So this endopeptidase can cleave within the peptide bonds. Okay, they cleave within the protein chain, peptide bonds within the protein chain. But there are some carboxypeptidase. It removes amino acids from the end of the peptide chains. Like if we see a structure of an amino acid, we will see that 4 groups are attached. H, R, NH2, one amino acid is attached and one carboxylic group is attached. Okay, so that is why every amino acid is having that 4 side. Okay, so it's these bonds are attached, but which side will be remain free? That COO minus and NH3 plus. So that is why this terminal is known as amino terminal, and this COO minus, this terminal is known as carboxy terminal of a protein. Okay, so this is a carboxy terminal, and these carboxypeptides work or cleave the amino acid from the carboxy terminal. So it cleaves this bond and separate this amino acid. Okay. So here is the activation process of the enzymes that are secreted from the pancreas. Okay. So firstly, enterokinase, it is present from epithelial cells of the small intestine. Small intestine is composed of epithelial cells, and that epithelial cells secretes enterokinase enzyme, okay, which activates trypsinogen to trypsin. So trypsinogen is the inactivated form and trypsin is the active form. So trypsinogen to trypsin, there is a enteropeptidase, that is the enterokinase. Enteropeptidase also similar with enterokinase. So this trypsinogen to trypsin occurs by this enterokinase, which secretes from the epithelial cell of the small intestine. And then trypsin, it's very important. When trypsin get activated, it activates the other enzymes like chymotrypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase, and proelastase to elastase. So it's very easy. So firstly, pancreas secretes trypsinogen, chymotrypsinogen, procarboxypeptidase and proelastase. After, when it's reached at the site of small intestine, enterokinase activates this trypsinogen to trypsin, and then activated trypsin activates chymotrypsinogen to chymotrypsin, procarboxypeptidase to carboxypeptidase, and proelastase to elastase. Understand? And see, functions of enzymes. Trypsin and chymotrypsin, these break large proteins into smaller proteins. Okay. Elastase breaks down elastin and other proteins, and carboxypeptidase cleaves peptides into amino acids from the carboxyl end. Okay, now you know what is the carboxyl end, carboxy end and the amino end, right? So you can see this chart, it will also very helpful. So within stomach, parietal cell secretes HCl and chief cell secretes pepsinogen, and this transformation pepsinogen to pepsin, because pepsin is the activated form, this activation is triggered by HCl, acidic condition. Okay, general protease with preference for acidic and aromatic amino acids. You can skip this point. Now the exocrine pancreas secretion into the small intestine. So pancreas secretes trypsinogen, chymotrypsinogen, proelastase and procarboxypeptidase. All enzymes are in the inactivated form. So only trypsinogen, chymotrypsinogen, proelastase, procarboxypeptidase, all are in inactivated form. They cannot cleave the protein now. So how they will get activated? By trypsin. And how trypsin get activated? By enterokinase. So where is the enterokinase? Enterokinase are present within the wall of the small intestine. So small intestine, wall of the small intestine secretes enterokinase, which converts trypsinogen to trypsin, and then trypsin also helps in the conversion of chymotrypsinogen to chymotrypsin, proelastase to elastase. All trypsin is only active form that activates the other inactivated forms of the enzymes to its activated form. Okay, and one more important point, that trypsin, it cleaves the amino acids, it select, because there is a long chain of amino acids. So which amino acid will cleave by the which peptide bond is cleaved by the trypsin. So trypsin is only specific for arginine and lysine. So it will scan the protein chain, that where is the arginine and where is the lysine, then they cleave that peptide bond only. Okay, and chymotrypsin, similarly, it will find out tryptophan, phenylalanine, tyrosine and leucine. That segment they will cleave, they will work on that peptide bond and they will separate from that point. Okay, and elastase will find out alanine, glycine and serine. Okay, so these are the tips and tricks for protein digestion. So now is the interesting point, that pancreas secretes that digestive enzymes, and they are very important and also very dangerous as they digest the protein. So, and I have said that our body compartments also made up of proteins. So there will be a high chance that our pancreas also get digested by its enzymes, right? Isn't it? Pancreas is holding that protein digesting enzyme, and how can pancreas get intact form? Pancreas produces a trypsin inhibitor, oh my god, to prevent premature activation of trypsin within the pancreas. That means pancreas is very clever. Pancreas is also housing trypsin inhibitor who keeps trypsin in inactivated form when it will present within the pancreas. So within the pancreas there will be no activation of trypsin, so protecting it from autodigestion. Important in preventing acute pancreatitis. That's my point. That's why pancreas do not digest, because it is having trypsin inhibitor. Okay, so what is the trypsin inhibitor? That is SPINK1, pink one. What is pink one? Serine protease inhibitor, Castle type 1, also called pancreatic secretory trypsin inhibitor, PSTI, inhibits premature trypsin activation inside the pancreas and prevents autodigestion and pancreatitis. Okay, so it's very important, right? So here is a clinical note that mutations, if mutation occurs and PINK1 gene are associated with an increased risk of hereditary pancreatitis. So PINK1 is its action, its function is to inhibit trypsin. But if it get mutated, that means its function get altered. It will not inhibit trypsin. So there will be high chance of hereditary pancreatitis, as it is linked with this PINK1 gene. That is why hereditary. Okay. So firstly, we have seen digestion occurs within stomach in presence of HCl. That pepsinogen is secreted by the stomach by its inactivated form. It's get activated in presence of HCl to pepsin, and pepsin act on the protein segment and peptide bond, and it cleaves into polypeptides, right? After that, there will be several enzymes that is secreted by the pancreas by its inactivated form, and that get activated at the small intestine site, right? Now we come to the brush border site of the small intestine. There are several enzymes present at the site of brush border of the small intestine. Okay, so final stage of protein digestion, it is the final stage of protein digestion occurs at the brush border microvilli of enterocytes in the small intestine, and brush border enzymes are involved like aminopeptidase that removes amino acids from the N-terminal of peptides. So who cleave amino acids from the C-terminal, that is the carboxy terminal? Yes, we already know that the enzyme name, that is carboxypeptidase. So carboxypeptidase can cleave amino acids from C terminal, then the N terminal will remain intact. So we also want an enzyme who can cleave amino acids from its N terminal. So that is the aminopeptidase, and where it is located at the brush border site of the small intestine. And there also dipeptidase, who splits dipeptides into individual amino acids. Tripeptidase breaks into small amino acids. Okay. And here is the function that converts small peptides from pancreatic, converts small peptides into free amino acids, dipeptides and tripeptides, ready for absorption. Okay, so here is the picture. It is the site of brush border where we are these are the microvilli, and there are the enzymes like aminopeptidase, dipeptidase and tripeptidase. Okay, so dipeptidase will cleave and separate the amino acids. Tripeptidase will cleave the peptide bonds and separate the amino acid, but only aminopeptidase, it will act only on the inside N terminal of a amino acid. Okay, so absorption of protein, it's very easy. Absorption of protein takes place within the enterocyte. Okay, and this is the apical surface of the enterocyte. Apical surface of an enterocyte, and there are presence of dipeptidase and tripeptidase, these enzymes, see, that are located. Dipeptidase, it will cleave the dipeptide like so. It will cleave this bond, and it will separate these two amino acids. Okay, next, here is aminopeptidase. This aminopeptidase, so it will act on that side and it will make separate like that type, one dipeptide and one free amino acid, right? So that is why here is generated free amino acids, dipeptides and also tripeptides. Okay, so there are several transporter that transport this free amino acid, dipeptides and tripeptides to the liver. But before going to liver, it has to pass through these transporters and enter through these enterocyte, and then it has to come that on blood, and then through blood it has to go to this portal vein to liver. Okay, so that is the route. So how they will transport? There are several transporters. We will see the name of the transporters. Okay, first transporter is located here. This one is the transporter 1, that is Na plus H plus antipode system. Okay, so this is Na plus H plus antipode system. If it is a cell, so outside of the cell is Na plus is high, that means here Na plus is high. So, and within the cell H plus is high. So Na plus will try to enter within this enterocyte. That is why I am writing Na plus here. So Na plus will enter to the enterocyte. Okay, Na plus will enter to the enterocyte, and H plus leave the enterocyte. That is why this system is known as Na plus H plus antipode system. Okay, Na plus H plus antipode system. Na plus H plus antipode. We know this type of transport. One molecule is going in and one molecule is going out. So the direction of the molecule is not same. They are moving in opposite direction. That is why this system is an antipode. Na plus H plus antipode. Now what is the next? That is that H plus now becomes high in the ECF. This is the ECF, extracellular fluid, and this is the ICF. Why H plus is high now? Because H plus is moving out from the cell and Na plus is going in. Okay, so H plus will go inside with this transporter within the cell. Okay, and who will move with this H plus? That dipeptide. So dipeptide will also move into the, so H plus, and who will move? Dipeptide. Okay, next for tripeptide it will also take help of H plus. So H plus and dipeptide, this is a symport because the direction of the molecule is same and there will be a symport. Okay, that tripeptide TP also enter with that H plus. Okay, but still there is so many Na plus available. There is so many Na plus available at the ECF, right? So these Na plus will also try to move inside the cell. So it will use this transport system and move inside Na plus, and that free amino acid will enter with Na plus. Okay, so Na plus also help free amino acid to move inside the enterocyte. Okay, so this one is 1, this one is 2, this one is 2, this one is 3, and this one is 4. Okay, and which one is 5? So now Na plus become high within the ICF, but Na plus concentration always high in the ECF, right? So we must remove this Na plus from the cell. That is why 3 Na plus will move outside the cell and 2K plus will enter the cell. Okay, and this is the function of sodium potassium ATPase pump. Na plus K plus ATPase pump, and that generate energy for this system to run. And there is a transporter, this one. From this, these dipeptide, see what happens. The dipeptide will move through this dipeptide into the blood. Okay, and this tripeptide will also enter through this transporter, and amino acid, free amino acid will also move through this transporter. So now they will lead to the portal vein and into the, this is the portal vein, and it will uptake all protein substance to the liver, and here absorption takes place. Okay, so once again recap. See, transport across the apical membrane into enterocyte. So di and tripeptide absorption we have seen. Transporter, name of the transporter is pepT1, that is a symporter. Mechanism, H plus peptide symport. Okay, and this is the secondary active transport. That means the energy which is liberated by the sodium potassium ATPase pump, these H plus peptide symport is using that energy. That is why they are secondary active transport. That is why they are secondary active transport. Okay. Next, di and tripeptides are co-transported with H plus. That means these two transporter, dipeptide with H plus and tripeptide with H plus, and these are symport and these are secondary active transport, because these two system 2 and 3, they are using the energy which is liberated by sodium potassium ATPase pump. Okay. Next, so H plus gradient is maintained by, now how H plus gradient is maintained? By Na plus H plus transporter system that exports H plus out in exchange of Na plus, that we have seen in this part. Na plus H plus antiport, Na plus H plus antiport system, right? Next, free amino acid absorption transporter mechanism, Na plus amino acid symport we have seen, that is also secondary active transport, and Na plus gradient is maintained by Na plus K plus ATPase, that is the primary active transport on the basolateral membrane, and which pumps Na plus out and K plus in using ATP. Okay, so that is the Na plus HK plus ATPase pump, and yes, this is the basolateral site. This is the apical site, apical border, and this is the basolateral membrane. Okay, this is the basolateral membrane. Okay, so intracellular processing, di and tripeptides are further hydrolyzed by cytoplasmic peptidase into free amino acids. Okay, transport across the basolateral membrane into the blood. We have seen that in this part, that when it crossed this membrane, this basolateral membrane, it mixed in the blood. Here is the blood, right? Next, this is free amino acid exist into the bloodstream via facilitated diffusion or Na plus independent transporters. In some cases, simple diffusion can occur. Okay, by means this transporter, this one, through this, when dipeptide, tripeptide and amino acid transport through this transporter, it is the facilitated diffusion or simple diffusion. Okay, where energy is not required. And see, so here it is the Na plus H plus antiporter. This is the peptide, this is the symporter. Okay, H plus and dipeptide and tripeptides enter through this peptide transporter, and NaH is the NHE is the Na plus H plus antiporter, and here is the sodium potassium ATPase pump at the basolateral site, and at the apical site there is the Na plus H plus transporter and peptide transporters. Okay, and dipeptide, tripeptide, amino acid, these transport through this transporter by means of facilitated diffusion into the blood, and then into the blood, and then from the blood it get uptake by the portal vein into the liver. Okay, so hope you understand this digestion and absorption of protein, and today's video is little longer, but hope you understand well with my simplest explanation, and stay connected with me. We'll see you later.